Hello, everybody, and welcome back to episode 10 of Daffy's Roundtable. My guest for today is a world renowned vertebrate paleontologist and evolutionary development biologist. I probably said that wrong. She is currently a professor at Carleton University and happens to be the professor I did my final project with there. I'm super excited for this episode, so without wasting any more time, please let me introduce my guest, Dr. Hilary Madden. Hello.
Hey, how's it going?
Good, how are you? Thank you for coming on.
Well, thanks for having me.
Yes, I'm very excited. I don't think we've talked about amphibians in depth this much till now, or hopefully till now.
Perhaps.
Yes.
Yeah.
Okay, awesome. So to start things off, can you tell us a little bit how you got to where you are now? The kind of research you're doing, that kind of stuff.
Sure. I mean, it's always a long story because I'm one of those kids that have always wanted to be a paleontologist. So
Okay,
my journey starts when I was probably about 4 or 5, but I'll skip
even
ahead a
better.
few years. Yeah, exactly.
Okay.
I was always one of those kids that loved dinosaurs. Jurassic Park was awesome. I was probably about 12 or 13 when it came out. So I was like, oh, my career is like real. I can do it. Went to university knowing exactly what I wanted to do, be a paleontologist. So was a terrible student until about third or fourth year when that was when I knew it had to be time to sort of step it up to get into grad school. Along the way, I kind of lost the love for dinosaurs. I think that was probably due to the fact that at University of Toronto, Mississauga, where I was, the paleontologist on faculty there did not study dinosaurs at the time. So studied instead Permian vertebrates, so animals that were living about 280 million years ago or so. And this was actually really quite cool because it was like a whole other environment, a whole other scene of animals before dinosaurs even show up in the fossil record. So kind of fell in love with that time period. And it's around there where we start to see the earliest reptiles, the earliest members of the mammal line, and our earliest sort of amphibians as we start to recognize them today. So I kind of became captivated by amphibians. I think their fossil record is super fascinating. In the past, they looked nothing like today. They were pretty large, fairly terrestrial, sort of reptile-looking animals, and over the evolutionary history became quite small and diminutive like the ones that we recognize today. So I kind of thought this transformation seemed to be a really cool event in the evolutionary history of vertebrates. And so that's basically what I studied now today.
That's really awesome. I actually had a few questions about the size change and whether they actually were much larger back then, because there is still the Chinese giant salamanders, I think they're called. Are they really as big as people? I've never seen one. Are they really as big as people say? And are they considered the oldest existing amphibian then because of their size? Does that make sense?
Yeah, it makes sense. I know what you're getting at. Yeah, so they are very large. So they have them on exhibit, I think, at the Prague zoo, and I got to see some of them, which were like incredible. So they're like 3-4 feet long, and awesome. They're just like exactly a flashback to what we think that some of the the Permian and age sort of amphibians probably would have looked like body size wise. But of course, like the anatomy is completely different inside the animal. So in terms of where they sort of reside in the family tree of amphibians or of salamanders, those animals are close to the base, but I don't think that necessarily means the ancestral or most primitive salamander was large, because another very closely related primitive group is very small. So I think that the body size growth was probably something that happened in the lineage that's alive today.
Okay, very cool. Okay. All right, so we'll get back to all the lineage and the timeline later on. Just a little bit more about like what you're doing currently with your research at Carleton. I know you're working with a lot of like axolotls and Xenopus. Maybe briefly break down what kind of research or what the research is looking for, I guess.
Yeah, exactly. So what we're doing is we do kind of like 2 lines of investigation in my lab. So, half of us are kind of working on building sort of the patterns of evolutionary change based on the fossils. So we're working on the earliest relatives of a lot of these lineages, trying to trace out what they look like and what those family trees of these animals look like. So we can actually start to see how the shapes and the anatomy has changed over time. And then when we kind of look at those patterns, we wonder, well, how did those changes come to be? What happened in the evolutionary history of those animals that sort of resulted in those changes that we see? And that's where we use the animals Axolotl and Xenopus to basically start to sort of test some of those predictions or hypotheses that we have. So did the expression of this one particular gene change in development such that it gave rise to a different kind of anatomy or morphology in an animal that we can basically manipulate today? And then in a sense, we're kind of seeing if we can replicate any of these transitional fossil forms in the lab.
That's very cool. And when you say manipulate, you mean actually manipulate the gene to do something else?
Correct. Yeah. So there's, some of our earlier studies, we were sort of basically exposing embryos to chemical agents that have known effects on the genetics of particular genes that we were interested in looking at. So we basically just drop the embryo in a little chemical treatment, take it out after it's done and let it grow and see what happens.
Very cool. Very cool. That's very cool. And then would you consider that the most exciting part of your job, or is there something that's even more exciting than seeing the changes after a couple of months or
That's pretty close. It's pretty close to being like the most exciting part, just sort of confirming that, yes, in fact, our predictions that came from rocks that were 300 million years old kind of came true in our hands. So, I mean, of course, it's also super exciting to discover new fossils in the field. So that's kind of where we have our aha and yippee moments over in fossil land. But yeah, if we can get an experiment to kind of confirm what we were thinking and what we thought was happening, that's pretty awesome. And you could probably attest to the screams of joy in the lab that have happened a few times.
I definitely can. I've been some of the screams of joy.
Exactly.
Yeah, that's awesome. Okay, and then about the finding fossils. So when you go out, are you mostly looking for amphibian fossils as well? I know I've seen you bring in like gigantic tree stumps. Are you looking for fossils within the tree stumps, or is that just a completely other project?
Yeah, no, that's exactly it. So we're looking for fossils in the Carboniferous era, period. So basically, where we know a bunch of these early reptiles and amphibians are evolving. But so when we go out, we basically just find whatever we find. So I've been hoping to find a nice clear record of amphibians, but as a result, we do also find a lot of reptiles and other kinds of fossils too, which are super cool as well anyways.
Of course.
And just what's special about Nova Scotia, where we collect, and all that material goes back to Nova Scotia, of course, but where we're collecting there, the way that fossils are preserved there is quite unique. So Nova Scotia is one of the only places where we can find vertebrate fossils of the Carboniferous age. And we find most of them inside those tree stumps that you've seen in the lab. And they're trees as in they're basically tree-like plants, but they're not related to trees like today. So they're basically more closely related to club mosses, very small plants that are alive today. But in the Carboniferous, these were 10, 20, 30 meter high tree-like plants. But also because they're club mosses, they didn't have like a woody core. So when the plant died, it basically would have snapped off, hollowed out, had a sort of a bark-like cylinder, and that would have been basically just an open manhole in the Carboniferous. So either things were living inside of there, got trapped and buried and preserved, or things got washed in and henceforth still got trapped and preserved. So we find most of the fossils, because they are so tiny and delicate, inside those tree stumps. They kind of were protected as a result. Yeah.
You kind of preserve better than in other areas.
Yeah, these things are tiny. I mean, you've seen some of the fossils in the lab. Their jaws can be, millimeters. Some of them are bigger. We're finding bigger and bigger animals, but a lot of these. Oh yeah, a lot of these earliest reptiles are, maybe 10, 15 centimeters. So it's only because of that really cool preservation that we have them at all, probably.
That's a chance. So is that why you do your field work in Nova Scotia? Is there any other reason that makes that have this written down? Like, what's the significance of the Joggins cliffs at Nova Scotia?
Yeah, the significance is basically that, yes, it's one of the only places on the globe that preserves vertebrate and tetrapod fossils of that age. And it's actually the oldest of those localities that preserves basically the first amniote, so egg-laying vertebrate, which also happens to be a reptile. So it's the first reptile as well. And it also has the earliest record of a synapsid, which is the animals that go on to become mammals.
Very interesting. And now that amniote, you released a paper on the first parental care in them, correct?
Yeah.
So those fossils were also found at Nova Scotia?
They were found on Cape Breton. Yeah. So slightly younger rocks, so not quite as old as Joggins. So
Okay,
if we had found that at Joggins, it would have been like a
incredible.
much, even earlier than we expected to see parental care. But as a result, still in New Brunswick or in on Cape Breton, it was still about 40 million years earlier than the previous record of any kind of parental care behavior.
So that's the earliest that we now know of in parental care behavior in just reptiles or
It
in
seems to
all
be in,
species.
we were trying to find other occurrences and records. It seems to be the earliest record for at least for amniotes, but it quite
Okay,
possibly for all tetrapods as well. So, uh,
very cool.
yeah.
That's yeah, very 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 cool. Awesome. Okay, and then back to the axolotls and xenopus.
Mhm.
Why do you use them as the like model animals in your lab?
Right. So we're using them right now as sort of like a starting point, just because my lab's not too old. We set up about 6 years ago, and so they
Awesome.
were currently known as model organisms. So there's a lot of knowledge behind them. A lot of the molecular techniques, like so, the genetics have been really well worked out for at least for Xenopus. The axolotl genome came out just a couple years ago. Um, they just have a lot of research behind them already. So starting out, it would have been the easiest to kind of jump into those systems. They
Very
aren't
cool.
necessarily the best model organisms for the kind of research we do, which is sort of the evolutionary research.
Right.
Because as you can imagine, if you think of an axolotl, it's not a very typical salamander at all.
Right.
so,
Yeah.
when model organisms are sort of boasted, sort of put as that status because they tend to be thought of as representing all salamanders. However,
Right. A big group of
axolotls,
them.
yeah, are not very good at doing that exactly. So we're actually in the process now of expanding our study organisms to include things like plethodon, the little redback salamanders from around here.
Very
They
cool.
do something very different from axolotls. They develop terrestrially. There is no larval stage, so they actually might be
Right.
better models for some of the earlier amphibians, which didn't have larvae. So
Okay.
we're starting to expand those tools into those animals.
So the earlier amphibians um, they didn't have larval stages. They didn't lay their eggs in water. They were first terrestrial, and then they started kind of going back and forth.
There's a lot of, as you can imagine, the fossil record trying to find little squishy
Of
larvae
course.
is really challenging.
Of
But
course.
it seems that amphibian-like metamorphosis is something that may have happened quite a bit closer to the origin of amphibians. Other fossil groups of amphibians have their own kinds of metamorphosis and sort of larval stages, but it's sort of thought that our modern amphibians sort of created this process as a way of dealing with interesting habitats. So we're not really too sure. I don't, they would have laid eggs in water at least because the amniotic egg doesn't appear till
Right.
later. But whether they
Right.
had sort of like a very distinctive larval phase like modern amphibians is still, I think, a little bit unclear.
Okay. Very interesting. And then, would you consider expanding past the salamanders into more terrestrial frogs and caecilians, for example? Or
100%, yeah. So we're
yeah.
we're working on bringing a frog, Erythrodactylus, Koki, the little
Oh, the
Puerto
Koi.
Rican. Yeah, so we want to
Yeah.
have those guys in the lab as well because they're also direct developers, so they have no tadpole phase. Um,
Interesting.
and as well, Bombina, the firebelly toad, great
Yep.
frog that's easy again to keep in the lab and is not as wacky as something like Xenopus. Do you have Bombina?
I have three of them. Do you want them?
Ah, are they? Do you know if they're male or female?
I don't. They're very young still.
Okay. Yeah, we'll keep
So,
posted on that because if we can
yeah.
get them breeding, I know there's protocols
Sure.
for injecting them for breeding like Xenopus. So
Okay,
yeah,
cool.
yeah,
I had no idea. I just like they happened to come into my care, and I was like, they're cool, but I'm not really like they're too young to be breeding or working with them yet. So I kind of
gotcha.
just
Yeah,
living here. Yeah.
yeah. I know. I
Cool.
worked with them a bit in my former lab. They're really cute. Yeah.
Awesome. Yeah, they are. I do. Have you ever worked with Spanish ribbed newts?
No, what's the genus name?
Pleurodellus
Okay.
waltel. What?
No,
Yeah. I'm from.
but I was wondering if it was pluralities. Yeah, because I know
Yeah.
there is a big community of developmental biologists who do work with that with that genus. So, but no, I think they're beautiful, but no, I've never
They're
worked
very
with them.
cool, and
Yeah.
my female is dropping eggs like crazy. So if you want to let me know, yeah.
So cool. Yeah. Oh, I'd love to take Sicilians in the lab too at some point. I've had them in the lab before. But of course, again, like being so enigmatic and cryptic, like there are not really well worked out protocols for even attempting to keep them for long periods of time, and breeding them is, is
Unimaginable.
is very rare. Is very rare. I think there's a
Okay.
few success stories around the world of people breeding them. I'd love to hear more. I'd love to have a contact that breeds Ichthyophis, any of the primitive ones, any of the rhenatromatids. Got any contacts?
Ichthyophis.
Ichthyophis, yeah, this one shows up on the pet trade fairly often.
I think. Let me look that up.
With the yellow band down the side, usually.
I think there is a. Oh yeah, I think there might be somebody called Nick Stacy in the States that's working with them. I will reach out to him and see if there's a possibility, or if he
Yeah,
has
that'd be
babies yet or anything. Yeah,
yeah, super cool.
that'd be
Yeah,
very cool.
Typhlonectes
Okay,
shows up on the pet trade a lot, but they're again they're a viviparous species, so they give birth to live young. But
okay.
Ichthyophis lays eggs, so I would be curious to see eggs.
You would want to see that. Okay, cool.
Yeah.
Okay, well, since we're talking about Sicilians, I actually have a few questions on them in here. Well, first of all, why don't we ever hear about them? So you're saying they're very elusive. Why
Yeah.
is that?
Yeah, so I think a number of factors. I mean, like they're fossorial, so they're primarily living underground. Even the most primitive members of the group are, leaf litter burrowers.
Okay.
So they are hard to find. There's been a dedicated research group in London who's gone out into the field and had success collecting them, and it's literally
Okay.
like digging up, fields daily is all you
To
do.
find one.
Yeah, so it's a lot of work. Yeah, I think basically just because they've been so cryptic historically, uh, there's not been a lot of interest in trying to, it just seems like not a fruitful avenue of research.
Of research,
Like
of looking for them.
to look for them, there's, comparatively like very few species known as a result. And is that real or an artifact? We're not sure. A lot of them
Right.
live in difficult to get to places. There's pan-tropical, but through the center of Africa has been sort of historically, politically tough to get in to do field work. Um,
Interesting.
so I think there's just, yeah, logistically challenging, and as a result, they have sort of stayed off the radar for most people, and we find different ways to answer questions.
Yeah, no, that makes sense. When you talk to even amphibian keepers in the hobby, you hear them talking about frogs and salamanders and all those different, but I've never heard anyone say I didn't know what Sicilians were until I met you. Like there's
Yeah. Oh no,
nothing
exactly.
telling.
I didn't learn about them till university either. And then as well, I've had them as pets, and basically, like, you just own a tank of dirt.
Yeah,
But
yeah,
yeah,
yeah. You
so,
don't even
so
see them.
so
Like
you don't see them. I mean, Typhlonectes, the aquatic ones, they swim around. You can interact with them and enjoy them. But other than just like for the joy of like caring for this animal, you don't get
you don't
much.
see it.
You don't get that much out of it. Yeah, yeah.
Yeah.
So.
So when you were feeding them, would you like throw in food that would burrow as well, and then hope they would find it under the soil?
Yeah, pretty much.
Pretty
Like,
much.
I tried to condition mine because I was curious if it was how hard or easy it was to move in the substrate I provided for it. Like, we're just kind of like winging it. Like, I just gave them like a
Right.
mixture of soil and like coconut fiber to kind of, but I didn't know how easy or hard it was for them to move in that versus the prey item. So I just kept like a compost of worms at home, and I would just
Okay.
bring in like earthworms every few days and just drop them in the same spot. And the Sicilians stayed alive for years and the worms
Okay.
disappeared. So presumably, like,
It was working.
it was
Yeah.
working. Yeah.
That's awesome. And then you dig to the bottom, you find the whole world colony and the Sicilians just living with them.
Yeah, exactly. Maybe. Yeah. We would dig him up every few months or so just to make sure he's okay. And yeah, he was like,
Right.
yeah, I'm cool.
And then like how long do they get? Like are they tiny
Yeah.
as well, or do they get pretty long? No?
Depends on the species as well. So,
Okay.
like, there are miniature species that I don't think would get much longer than 10, 15 centimeters, but there are
Awesome.
species in South America that can get to over a meter in length.
Wow.
Yeah. So they
Wow.
can be quite big.
And they still exist? They
Yes.
haven't. Yeah. Wow.
Yep,
Okay. That's
yep,
very
yep.
cool.
Yeah, the ones like Ichthyophis, probably, like maybe 40, 50, 60 centimeters. Dermophis, some of the common ones on the pet trade won't get terribly large, maybe a foot or
Okay.
2 feet length total, but yeah, there's some big boys out there.
Yeah, I wonder if they get sold in the pet trade as Sicilians or if they get sold as other species, like maybe small snakes without people knowing what they are. Or
Yeah, I've seen, so because of the rules of exportation from places like Brazil, they
right.
get smuggled out, I would say, or they get taken out by being under the name of fish, rubber eels.
Oh,
Um,
interesting.
yeah, so
And that works for the aquatic ones.
yeah, yeah, yeah.
Okay.
I
Okay,
have
that's
no
very
idea.
interesting.
Yeah, I have
That's
no
that's
idea
I
how to
guess
get them.
that's also an easier way to find them. We could try to look for rubber eels and see if they, I guess,
Yeah,
maybe.
totally. That's how they'll be labeled on tanks and fish store, or yeah, fish
Exactly,
stores is like
yeah.
that too. Yeah, and you're like, wait, you're not a fish at all.
I'm wondering if, yeah, that's very interesting. Maybe I've even seen them then in the past and just
It's
been like, yeah,
possible.
fish, and then just
Hmm.
walked away. Okay, very interesting. And then, so do we know where they kind of fit on the historic timeline? Because they look very prehistoric, but I guess because we don't see them, we don't know the research as well.
Yeah, exactly. Which is why I did my PhD work on them. Like,
Oh,
my entire
interesting.
thesis was on Sicilians, trying to figure out what in the actual heck are they like? Because yeah,
Okay.
the fossil record of them is very, very poor. Like all of the fossils known could probably fit in my hand or maybe two hands, for
Wow.
Sicilians. So and they have a very unusual morphology today. Like the modern ones are really different from anything that looks like anything in the past. So we're basically
Okay.
trying to connect something that occurred. The oldest Sicilian fossil that we think is definitively a Sicilian is, from the Jurassic, and then the gap to where it may have branched off from goes back to the Permian or maybe even late Carboniferous. So
Wow.
there's a
So
huge, huge gap.
nothing in the middle,
No,
but there
no,
must have been something.
there must have been something,
There
but
must have been something.
were they living underground as well, and hence like did not get preserved well, living in tropical warm regions where things decompose quickly, they're very small. All these factors probably lead to them not having a fossil record. And so absolutely, as a result, there is a lot of debate amongst paleontologists about where they actually fit in the family tree. Are they closest relatives to frogs and salamanders, or are they derived from a completely different group of fossils, or different parts of the same sort of broader family tree of where frogs and salamanders fit? We're still, literally, I'm still working on this today. Like yesterday, my project was was trying to reanalyze some data and try to figure out a more robust idea for where they belong.
Interesting. And then, so you said your thesis was, what are they? Did you come to a sort of a conclusion or an assumption?
I did at the time, yep. So I gathered a whole bunch of new CT data, new morphological data of the inside of the skull, the brain case bones,
Yeah.
and reanalyzed new characters from that anatomy. And my hypothesis that emerged was the same as what a few other people were sort of leaning towards thinking and sort of building on a common story. However, since then, that was back in, like 2010, 2009, new fossils have come to light that have sort of shaken that up again. So we're back to
Back to
reanalyze
square one.
that. Yeah. So keeps
That's part of
us busy.
science, Maddin.
Absolutely. Yeah.
Yeah,
Every new piece of data changes our vision a little bit, and it's okay.
for sure. Yeah,
Yeah.
okay, it keeps. I mean, eventually, I guess we'd never run out of fossils. But if that had never happened, then there would be no more paleontologists at some point. Not now, but at
Exactly.
some
Yeah.
point.
And we're
Yeah.
not slowing down. Yeah. There's still good
No.
stuff
Yeah.
coming out. Yeah.
Is so? Are even like when you go out to like Nova Scotia or some other field sites at this point, do you find it harder now to find fossils than you did a few years ago? Like, do you feel like um, maybe the areas have
Mm.
already been discovered, so now you need to look in different directions? Or
No, not at all. The rate of erosion of those cliffs is high
okay,
enough that any given year we have
wow,
an equal or better chance of finding something than the year
wow,
before. Yeah. Yeah.
that's very cool. So
Yeah.
it's the erosion of the cliffs that
Yeah.
kind of reveals it. Okay, interesting.
Yeah. Yeah.
I Very cool. Okay,
Yeah.
and then can we talk a little bit more about axolotls for
Yeah.
the
Yeah.
other
Sure. Yeah.
species? So is how so everybody, even people who don't know what axolotls are, probably know that axolotls can regenerate certain limbs
Mm-hmm. Mm-hmm.
or most of their limbs, I believe.
Yeah.
And I've even seen gills as well, right?
Oh
So
yeah.
how is this possible, or why is this possible, or if there is
Yeah.
a simple answer for that?
There probably is if I were a regenerative biology scientist, but
Okay,
I
okay.
don't know. Okay, so I know axolotl can regenerate the parts of their heart, their
Oh
jaws,
wow.
parts of their brain. Like their capacity is unparalleled, right? So we've
So
never
there's
seen anything
no,
like this.
it's not just the limbs and the fingers, and it's,
No.
you're saying hearts and jaws. Wow.
Yeah, yeah.
And you're saying there's no other species that can do as much as that. Wow.
As far
Okay.
as we know, no, yeah, exactly. So other salamanders, other newts can regenerate little bits of pieces of themselves. Frogs a little bit, and then also often that's usually the higher capacity to regenerate is earlier in their lives. Like if as tadpoles, they can regenerate
Right.
parts that maybe they can't as an adult. Um,
Right.
I don't. I mean, I'm at risk of like totally like making this wrong. My understanding, as far as I can
No,
tell, is that salamanders, well, axolotls seem to have this ability to de-differentiate like wound cells back into
okay.
stem cells, such that they can then basically retransform what was sort of that wound site where you had already injured bone and muscle. They basically re-encapsulate that, de-differentiate those cells back into stem cells so that they can then regrow and be fated to
I'm,
become. And so that's exactly the mystery of what they're, regenerative scientists are trying to figure out, what is involved in that process that we are lacking
that,
as.
yeah, that, why, what allows them to do it?
Yeah, yeah, yeah. So I think we're not
Very, very
quite
cool.
fully understood in what those mechanisms are exactly, and then how they relate to what we possess and don't possess. But that's basically where that research is going. I mean, obviously, if it's a matter of just like giving us a shot of some gene product that allows us to de-differentiate cells, then
Yeah,
the
yeah,
they're trying to figure that out
yeah. It
intensively.
would be good
Yeah,
for injuries or something later on down the road. But
hugely.
we'll
Yeah,
leave that to the regenerative or
exactly.
whatever you call them.
Yeah.
Okay, cool. And then is with the what you guys call the GFP
Mhm.
gene, and would that also be something that they kind of do research on, or is that more um
Yeah, no, so the GFP is more
first of
like
all, what does it stand for?
yeah, it's a tool. So it's the green fluorescent protein
Okay,
transgenic of axolotl. So what they've done, what the basically the scientists have done is taken a jellyfish fluorescent gene and inserted it into the genome of axolotl right behind a part of a gene sequence, the enhancer that basically is expressed to sort of guide a gene that's expressed in every cell.
okay,
So one of these sort of like housekeeping genes. Again, I don't know all the details of the
yeah,
mechanics of this, but
no, for sure.
basically, what it means is that in order for that normal gene to be expressed in axolotl, the GFP has to be expressed before it, and then it continues on its way. So it basically, all those cells then produce this green fluorescent protein that then floats around inside the cell. It's harmless, it does nothing, doesn't affect the function, but when you shine UV light on it, it glows. And so we basically have this any of these types of transgenics are basically just tools for us to further understand something else about the animal. So what we use them for is for understanding what parts of the embryo give rise to what parts of the adult. And so we can actually graft pieces of the GFP positive, sort of a donor embryo cells into a normal background wild type embryo and grow it up and basically see whatever parts of that larva or juvenile salamander glow green. We know that that must have come from that part of the embryo.
That's very interesting. That's very, very interesting. So,
So when we start manipulating the genetics, we can see specifically what parts of the embryo's fate did we change.
and whether it's working or not, right? Like
Exactly.
if you
We can
don't see
confirm
the, if you
exactly.
don't see the
Yeah.
green glow, it didn't work. That's very interesting.
Yeah.
And then
Yeah.
is this a breedable trait? Because I, like, right now they're in the pet trade as well.
Yeah.
So
Yeah.
I guess maybe somebody bred an axolotl that was injected to a regular one, and now they've continued.
Yeah. Yeah. So the way they were constructed was that the sequence to create the protein, the GFP protein, was inserted into the genome of an original axolotl, probably a few. And so it's like a part of their genome now. It's in there.
Okay.
And so every time you breed them, it's going to be passed on to offspring. The way I think
Very
it
cool.
got sort of put in, at least at one facility, the way they put it in is that if you were to breed, I think it's sex linked. So if you were to breed a GFP and a leucistic, only half the embryos would be fluorescent and half
GFP.
would be, yeah. So that's actually helpful for our work because we need sort of synchronous donor and host embryos, so we can move
Right.
pieces between them instead of having like trying to coordinate two sets of breeding. We can do one breeding, get embryos that are GFP and non-GFP, and do our experiments that way. So yeah, it's in the genome. Anyone could breed two GFPs, get GFP babies. Yeah. That's how it
Very
would work.
interesting. And is this in other have they done this in other amphibians, or is this only in
Yeah, I have GFP. My Xenopus, half of them are GFP. You just can't see it because their skin is so pigmented.
right
So it's not as obvious as obviously, you can see in
in
an
the
albino
axolotls?
or a leucistic axolotl. Yeah. Yeah. Oh, there's GFP mice, chickens, rabbits, pigs.
Chickens.
Oh yeah, yeah.
How,
Any
how do
animal
they
that's sort
see
of
it in chickens?
under a UV light, you can excite it as well. You can see it in rabbits, cats. They've genetically
Ah,
modified all these species for research to have this tool available. Yeah.
very, very cool. Okay. I, yeah, I'm sure. Like, I did know that it existed, and I've obviously seen it before in your lab, but I didn't
Yeah.
know that it wasn't that many animals, and that it was. Um, I don't know why I assumed at some point it might have been like a natural genome that popped up, like in jellyfish, and then somebody had taken
Right.
that and kind of separated it. But I guess
No,
it's
yeah. Well, because that's the story with the albino axolotls, right? Like that was just sort of a fluke occurrence in the
s-
wild, and they collected them and have bred every subsequent albino axolotl out of like the same seven or something.
yeah, it's the
So,
same thing with the leucistic Spanish ribbed newts. There was just
uh,
one breeder who got like a one batch with like three or four of them, and now every single
yeah.
one is from the same, which also questions, I guess, inbreeding and how
Yeah.
healthy they are down the road. But
Yeah.
um,
Totally.
awesome. And then so I guess I have a few questions here about like amphibians first starting out.
Hmm.
Um, but I feel like you kind of already busted these myths for us. So amphibians weren't the first animals to walk on land, right?
No.
No. What would have came before them?
Well, I mean, when we're in Nova Scotia, and as well in neighboring New Brunswick, we're seeing some of the first terrestrial, like fully terrestrial vertebrates. And so these are these earliest amphibians, earliest amniote-like, reptile-like creatures. Um, then they're 320, 330 million years old. Then there's trackways that are even older than that. So 380 million year old trackways that we just don't have body
Trackways, like
fossils, as in like footprints.
footprints. Okay.
So we
Wow.
can
Okay.
see their footprints. There was animals on land, but often where we get footprints preserved, we don't always have conditions that preserve body fossils. So we just don't see
Right.
the actual animal fossils, but we have evidence that they were walking on land like a modern amphibian or reptile.
Four legs as well. Like
Four legs as well. Yeah, most 5 digits, 5 fingers, 5 toes. The earliest sort of limbed vertebrates had more toes and fingers than that, but the first really definitively terrestrial animals had you know limbs just like ours. Yeah.
that's very interesting. And then what's the relationship with the limbs? So I
Mm.
know I've seen a lot like you do research on like the number of digits between this like what the final project was, but like
Oh
what's the
yeah.
relationship of having, let's say, this species having three and then that species having four digits? Like how do you kind of link that that's the common
Yeah.
ancestor? Like does that
Right.
question make sense?
Yeah, I think it makes sense because okay, so I think what we're when we look at the fossil record, pretty much the vast majority of animals have 5 digits on their hands and feet. So that sort of
Right.
like is the most common condition for these earliest terrestrial vertebrates. It's
Okay.
not until animals start experimenting with different ways of living, like being more eel-like, or for burrowing, or for becoming secondarily aquatic, where we see limbs starting to be reduced again, and often in those cases, that's where we start to see digits dropping off the
Oh,
animal. So, I mean, salamanders, a lot of them have lost at least one digit or two on the hand.
A lot of three-toed salamanders, yeah.
Yeah, exactly. So, yeah, and those are just sort of unique occurrences within those lineages for whatever reason they were adaptive or not maladaptive. So whatever happened, they're
So
just
like
repeated that
either
way.
they needed them to be three or they didn't see a use for having the five,
Yeah,
right?
yeah, yeah. Or maybe,
That's
yeah, they might be in the middle of transitioning to like losing their forelimbs altogether, or
altogether.
losing a
Okay.
limb altogether, which is what we see in a lot of squamates that are limbless, secondarily limbless. We see sort
Yeah.
of transitional forms in between, which is what we were sort of trying to figure out with your project. Like, how common is that? What are the patterns of limb loss and digit reduction and whatnot throughout that group?
Right. So when you, for example, see like a ball python with like the one digit, they have like, so would that be considered like the through the process of losing the digits?
Yep,
Or
yep,
yeah,
yeah, exactly, yeah, yeah,
very
yeah,
interesting.
yeah.
Very interesting. Awesome. And then so we already discussed that the Chinese giant salamanders weren't the oldest. Do we know what the oldest currently existing amphibian is?
Oh, like when it appeared in identifiable form in
Y-
the fossil record?
y- Yeah.
Yeah. I'm not too sure because I don't work on many of the young ones. Like, I mean, I consider
Right.
like Cretaceous aged amphibians to be young, like it's
Right.
only 65 million years, or it's like 80 million. But I
Not
think,
old
yeah,
enough.
not old enough, no. But I feel like a lot of recognizable crown group, at least families that have living members in them today, appeared at least, in parts of the Cretaceous. The probably some amphibian biologists are freaking out at me saying that, but my understanding is a lot of groups had started to originate by at least the Cretaceous, and then, yeah, maybe a bit earlier or later. I'm not too sure. Yeah.
Okay. In that case, what are some of the species like the old ones that you are kind of finding in the fossil record?
Mm-hmm.
And then maybe I can throw up a picture on the screen.
Right.
Like there's one, I seen one that a lot of people on campus always have on their shirt. And it
Oh,
has like a triangle head, maybe.
yeah, Diplocaulus. Yeah.
So, but
Yeah.
that's not what you work on though.
I would love to. That's a whole other group of animals that I haven't even gotten into. Yeah, those are members of like Nectridia,
Okay,
a group of currently thought of lepospondal, temnospondal, or tetrapods. But I primarily work in another group of fossils, the temnospondals. But again, there's arguments. Maybe they belong over another part of the tree. They're just another weird group of amphibians that we don't know much about. They have no living
right.
descendants, as far as we can tell. So just they went completely extinct. In the Permian, probably.
Very interesting. What is the word you just used?
Oh, like temnospondals?
Yes. What does that mean?
Yeah. So that's a name for a group of tetrapods that includes most, if not all, of the modern amphibians. At least, it's their kind of the strongest hypothesis at the moment is that all frogs, salamanders, and Sicilians came from that fossil lineage.
Okay. Very
Yeah.
cool. And then other things would have come from the
Some other things, yeah, stemmed out of the lepospondal group,
Leopold.
and
Okay, that was yeah.
lepospondals are more closely related to amniotes, so they're just right outside of the amniote group. But
Okay.
again, if no amphibians are descendants of lepospondals, that entire clade went extinct as well. So we have no living descendants of that group.
Oh, at all.
No.
Very
So it's kind of
interesting.
sad. You're like, oh,
Yeah,
sometimes I
yeah.
wish like sometimes I wish some of the amphibians were lepospondals so that we didn't lose like an entire clade
An
of
entire clear. Yeah.
of diversity, but it is what it is, I guess. Probably end Permian extinction was the big bottleneck.
That's when a lot of them, a lot of the species got. We lost a lot of amphibian species
We did, yeah,
and the
yeah,
Leospondylus.
yeah,
Okay.
yeah. A lot of the lepospondals disappear. Yeah.
Very cool. I think that's about all the questions I had to ask you, but I do
Okay.
want to ask you about mud puppies before.
Oh yeah.
Oh, I do have one more question, but mud puppies
Okay.
first.
Okay.
Why are they so hard to find? And then what makes them like? I know a lot of groups are doing research on them. So why are they so important for research? Is it solely because they're so endangered at the moment that they're kind of
Mhm.
doing environmental like research on it, or is there
Hmm.
kind of like a historic significance to research on them?
I'm not too familiar with too much research happening on them right now.
Oh, okay.
I feel like, I mean, so historically they were um, a lot of bycatch in like the Great Lakes and things like that,
Right.
which I think made them good models for teaching anatomy in
Okay.
schools, right? Like
They were there. Right.
we dissected my puppies. Um,
Right.
and I think just, yeah, with the sort of like crashing ecosystems around the whole Great Lakes region, I think they've become threatened as well as probably because of this over harvesting, maybe too. I don't know too much about their sort of their current status. I think though that's sort of my understanding is what some of the main drivers of why they are pretty rare and hard to find. I think they're fussy animals to keep in the lab. If we were to try to, did you come out did we see them? We never
We
saw
didn't see them
any.
when I went
Okay.
out. No. Yeah.
Okay.
Yeah.
But you saw like their habitat, right?
I
It's
did.
like
So it's very fast flowing water, and
exactly.
that was actually another question. So so a lot of the salamanders and like the newts and I guess the aquatic ones, so the newts and axolotls, they all seem to stay in very calm, slow water, and I think that's more
Right.
to do with like the way they swim and the way they move. But
Yeah.
mud puppies were in super fast water, like that was like a waterfall. So
Yeah. Yeah.
it does that, yeah,
I
like
think
is
that's
there
probably why it's hard to get them in the lab. I mean, I think they need like flowing water over their
constant moving water,
gills, like high oxygen environments.
right?
Um, maybe I honestly, I'm not even too sure. I think they're just
Okay,
very cryptic. They seem to be very sensitive to environmental perturbations, so they're not found everywhere. They're only found in sort of these very unique kinds of places, like the place that we go to near here.
yeah.
It's the only kind of creek that has them like that, like nothing else
In the
around.
entire area.
Yeah.
Wow. Okay.
So, and whether that, I think there's arguments about that is partly due to the fact that they built a little dam on that creek and it actually moderated the environment so they couldn't go further up, so they basically are sort of
Okay.
trapped in this sort of
Trapped
zone
there.
where we can find them. But yeah, I think they're just over harvested historically, probably have bad environmental conditions now and have never really been protected because they're pretty cryptic.
Yeah.
And so now I think are in bad shape. Yeah.
As most amphibians are pretty cryptic, I think, and which is why I find them so interesting. I think it's the ability to survive in such weird places, but still like not ever be seen.
Totally. So cool.
Yeah.
I mean, these neotenic salamanders are so cool, right? Like they're doing something like nothing else does. So
No other species
yeah,
can do.
yeah.
Yeah, absolutely. And they are completely like they never. There's no. They are like axolotls. They never. Their entire life stage. Do they have a larger stage
No, I know. I mean, like they're larval in that they're just smaller versions of
of
themselves.
themselves? Okay.
There is a metamorphosis that happens. Their blood will metamorphose like axolotl, and parts of their tissues metamorphose, but you
Very
don't
cool.
see that in their external features. They will maintain those
Okay.
larval external features. Yeah.
Axolotls' bloods metamorphose.
So I think there's like there's blood cells that metamorphose and change. Like
That's
there's weird things that happen to the different tissues that are like cryptic to our eyes, so that we can't tell that they are actually mature visually. But yeah, there is sort of like a very cryptic metamorphosis that does still happen in a sense.
See, the more you talk about amphibians, the cooler they get.
I
Like it's that's very cool.
exactly. Yeah. Yeah.
Awesome. Okay, and then finally, my last question, and then it's the most important question of all. What is your personal favorite amphibian?
Oh,
I know it's a tough one, but
it's tough. I mean, I think like obviously things that come to mind are like glass frogs. I think they're so cute. I mean, it's
yeah.
probably going to be a frog of some sort. Their
Okay.
eyeballs, I just can't handle how cute their eyeballs
So
are.
frogs, not the salamanders or the Sicilians.
Yeah, I mean, if I were
I
to
will
like
leave it at we.
choose, yeah, if I were to choose one to live with for the rest of my life, I think I would probably be a frog. I think
Okay,
they're
sweet.
just super cute. Yeah.
Yes, as a dart frog keeper, I have to agree.
That's so
Yeah,
cute. Yeah.
they're very very cute, and they the hops too, right?
Oh, and when the ones that crawl, like the little like toads, the little mini toads. Oh man, yeah.
Yes, no, agreed, absolutely. Awesome. Okay, So that was everything I had written down. I kind of wish I had more written down, but because it
Oh,
was so
that's all right.
interesting. But anyway, we're already at where I usually like to keep it around 45. So
Cool.
that's perfect. Thank you very much for coming on. I'm super excited to post this episode.
No problem. I'm like, oh man, I like butchered so much random stuff.
No, I mean, no, I'm sure that's not true.
We'll see.
Awesome. And then, so I usually tell people to like say where people can find them at the end of the episode. So are you on any social media that you want people to maybe keep up with you on, or
Yeah, I guess on Twitter, like, I mean, that's where I
Twitter?
kind
Yeah.
of post most of the research stuff is, yeah, I can find me at Evo Diva. 'Cause it's
Evo
funny.
Diva.
Yeah.
Awesome. Yes. I love that. Okay. I will also have it in the show notes
Okay,
as
cool.
well. Perfect. Thank you very much once again.
All right, no problemo.
Talk to you soon.
Okay,
Bye-bye.
bye.
Awesome. Thank you very much once again to Dr. Hilary Madden for coming on the show. That was very interesting. I learned, I mean, I've been in and out of her lab for almost 3 years now, learning about amphibians, and I still feel like I learned so much on this episode. So hopefully you guys felt the same way. So follow her on Evo Diva on Twitter. Follow me on Daffy's Reptiles on Instagram, YouTube, everywhere else, and then drop this show a like and a subscribe on whatever platform you use to listen to podcasts. Thank you very much, and we'll see you on the next one.